Nelson specializes in the fields of mycology and aerobiology. He has practiced environmental consulting since 2005. He was an environmental microbial analyst for several years and is one of a few environmental fungal identification experts in the state of Arizona. He has a master's level scientific background with foci in mycology, plant pathology, and forest ecology. He presents Fungal Ecology lectures and guides mushroom collecting forays in northern Arizona. Erik's forest pathology research currently supports important forest health planning models used by the U.S. Forest Service. So, before I do the really big welcome, please remember to silence your phones and don't talk during the presentation. For this one, we're not saving Q&A to the end. We want it to be more of a conversation, but give Alex or I, whoever's got this mic, to kind of come to you when you raise your hand so that people online can hear you. We do not have a Zoom live stream today. No, I don't know. Oh, no, we have Zoom. We don't have a YouTube live stream today. I have no idea why. It's just the problem of the event. Always have to have a problem. And so I'll be posting this recording later on YouTube. But we do have Zoom speakers here today. Or Zoom speakers, attendees. Yeah, there's a lot going on in this head right now. So, everybody, please join me in welcoming my favorite mycologist, Erik Nelson.
Thank you, Catherine. And an extra round of applause for Catherine, who made all of this possible. I'm honored to be here with you today. I would feel a little bit better if I had the screen back. But yeah, I met Catherine through an atheist meetup group, and we played a game of Wingspan together, and that's how all this began. I am one of you. I'm a strong advocate of secular humanism myself. Is anyone here aware of the BASIS schools, the college prep high schools and that called BASIS? I taught for 1 year at BASIS, and if you're familiar with the schools, they have an 8th grade required course called Logic. Can you believe that? So a whole semester, I got to instruct 8th graders, which mostly was discipline and got 20% content. Mostly taught 8th graders critical thinking, logical fallacies, philosophical logic, all the good things that really should be core curriculum throughout the country. Our world would be very different if everyone had critical thinking skills from the get-go. So I'm honored to be here. Thank you so much. I'm happy to be in good company. So the plan for this talk, I'm going to keep going until we have a display. Go ahead. All right. So the plan for this talk, I have three sections to this presentation, and I'm sorry to contradict you, Catherine. I typically have more time, and I am more back and forth with the audience, but we're limited in time, and I have a decent amount of content, so this is going to be more lecture-like. I'll involve you as much as I can, but I think we're going to save the major questions for after the presentation. So the three sections of my presentation are, first off, just what are fungi? What makes them unique? And then how the theme of the presentation, by the way, is mushrooms are treasures. How are they treasures? Well, for the ecosystem, they're incredibly valuable. I'm going to cover some of the major aspects that fungi play in their ecosystem and why our world works because of fungi. And then secondly, we're going to look at the human aspect, why people like mushrooms, are interested in mushrooms. So let's begin with all life on Earth. So let's say that we were only given 5 categories to place all life on Earth in. What would some of those 5 categories be? Who wants to give a shot? Just shout it out. Animals? Prokaryotes, very good. Do we have a fellow microbiologist in the group? Or okay, all right, good. Prokaryotes, excellent. Fungi, there we go. Any other takers? Archaea, excellent. That would be with the prokaryotes, very good.
What's a prokaryote?
I will describe. Trust me, it's coming. All right, we're good. Any other takers for any of the other groups? Go ahead.
Eukaryotes.
Say again. Very good, eukaryotes. Of course, we divide all life, if we were to only have two groups, we would divide all life into two groups, the prokaryotes and the eukaryotes, which I will now describe. So good job, you got most of them. Those of you that said eukaryotes may have been thinking of the protozoans, the protist kingdom, which are single cellular, but everything that's not a bacteria on this list is a eukaryote, and I'll explain what those are. So here's our wonderful tree of life. And here's the division between prokaryotes and eukaryotes right here. So most life on Earth came from the more complex eukaryotic life that is not bacteria or archaea, and we'll talk just a little bit more about that. The thing I love about cladograms and the tree of life, which is a form of a cladogram, is these branches mean something. This means echinoderms, which are the starfish, evolved after and from a common ancestor of all of the things that we typically consider animals, reptiles, fish, birds, etc. They share a common ancestor. So what I like to point out is that when the protozoans, the eukaryotes first started to develop, as soon as they became multicellular, we had a branching where the single-cell protozoans continued, and the other branch that came out was probably single-celled algae at that point and developed into plants. Later on, we had a branching between the fungi and all the other things that we would call animals. So animals and fungi have a common ancestor. We are more related to fungi than the bacteria or the plants. And fungi are more related to us than they are to plants. So it's been a while. It's been about 10 years, but I was on ABC 3 TV talking about poisonous mushrooms in the valley. And how did they introduce the piece? There's a dangerous plant popping up all over the valley, right? So these things still exist. It's probably the high school teacher's fault. But these things, these misconceptions still exist. In fact, fungi is called the fifth kingdom of life because it took quite a while. I think it was around the 1950s, 60s before we actually recognized fungi as a separate kingdom of life. So I like to bring up the tree of life to kind of point that out. So let's just do very briefly, I do have limited time, very briefly on these different kingdoms. So bacteria, these are the prokaryotes. They have very simple cells. They do amazing things, and their place in an ecosystem can be very complex. But their cells are fairly simple. They don't have organelles. So all the eukaryotes are more complex. They have things like mitochondria and a nucleus and my biology professor's favorite word, endoplasmic reticulum. Bacteria don't have these things, but they're amazing in that they power themselves by acting like chemical factories. Chemical in, transformation, harness the power of a reaction, different chemical out. So they play really important roles in, for example, the nitrogen cycle. Rhizobium that grows on the roots of members of the pea family, Fabaceae, contribute to the nitrogen cycle. And many of the plants here in our desert are pea family plants. Palo verdes and mesquites are nitrogen-fixing plants, for example. That's why they have high protein in their beans and in their pods. So I mean, obviously, each one of these slides could be an hour in itself, but these are the prokaryotes bacteria. I do have a story. I'll try to be quick on it. I recently did a project, so I'm an environmental health and safety investigator consultant. I recently did a project on a newly constructed campus down in the Yuma area. They were having problems with copper corrosion. They were getting a lot of copper in their water. Their water was bluish colored. They had all these blue-green mineral buildups in all their aerators. Their water smelled like rotten eggs. And we worked with them to figure out what was going on. Well, it turns out in Yuma, because of all the agriculture, one of the more popular fertilizers is ammonium sulfate. There's very high sulfates in the aquifer water that everyone pulls their water from. When we looked at, so we look at the water at different points along the plumbing line, and one of the most important areas to check is the water heater. Because if the water heater has problems, it can be a very wonderful place for bacteria to live. When we drained the water heater, did I take too long? Okay, I'll keep talking. When we drained the water heater, it smelled horrible, rotten eggs, and the effluent was black. It was terrible. So as an investigator, I took water samples all along the lines, and what we found was high bacterial loading in the water heater. And when we looked for, I ran a panel specifically for corrosion-related bacteria, and what do you know, what did we have really high results for? Sulfate-reducing bacteria. This is bacteria that powers its cells not with organic materials, but by taking sulfate itself and converting it into hydrogen sulfide. Hydrogen sulfide smells like rotten eggs. Hydrogen sulfide, when it comes in contact with water, creates sulfuric acid, which corrodes metal. So we got our culprit there, and that was because of the little chemical factory, sulfate-reducing bacteria, doing the work. All right, protozoans. These are some of my favorite forms of life. Single-celled, but these are eukaryotes. Everything here and after are all eukaryotes. Why? Because they have all these wonderful things. These are called organelles. They have the mitochondria. They have the endoplasmic reticulum. And all complex life that came after bacteria are eukaryotes like these guys. Protists are amazing because they're like little microscopic animals. They fill every ecological niche. You've got producers, you've got consumers, you've got scavengers. Every niche that there is in an ecosystem, microbially, they take, including parasitism like Giardia, Plasmodium, sleeping sickness, malaria, all these wonderful things that we love, right? And they're super fun to see under the microscope, of course. And so now we have the plants. And the plants are complex, multicellular life, but they also can exist as a single-celled algae. All plants came from algae, and in some ways, they can be considered just a more complex algae. So in the order of evolutionary history, we have the bryophytes, the liverworts, the mosses, horsetails, equisetum, ferns, and then we have the gymnosperms, which are the conifers, more than 500 kinds, conifer, juniper, and pine. The angiosperms, the flowering plants. And so what makes plants so important and so special? Who just wants to say it? Photosynthesis, very good. What do? They take carbon dioxide from the air. They take water from the ground, and they take sunlight. And they make, from carbon dioxide and water, they make carbohydrates. And everything the plant is all carbohydrate. And so one of my favorite scientists of all time, Richard Feynman, likes to say plants are made from air. And they really are. Where does the rain come from? It comes from moisture in the air. So everything that a plant is comes from the air. And then we have this incredible diversity and all this biomass from the air. So plants are, when we classify how things eat, what they live off of, plants are photoautotrophs, which means they use light to make their own food. Trophic means where you get your food from. So it's important to recognize that because everything after, like us, the animals, we are heterotrophs. We get our food from other things. We get our food from the organics, and plants are certainly one of these. And of course, because animals can outrun plants, we have pretty good advantages, and we can outrun each other too, so we also eat each other. But yeah, we're heterotrophs. We need to get our food from outside sources. We're all multicellular, and one of the things that defines animals is we have a digestive tract. Even this little microscopic hydra has a digestive tract all through there. So it's important to recognize animals are consumers. Animals also are fast, right? We can outrun plants. We're highly mobile, we're highly adaptive, we have a nervous system, we can think, we can communicate, all these wonderful advantages that animals have. Why is half of this screen dedicated to arthropods? Because arthropods dominate animal diversity. The insects specifically, the number of species in the insects is greater than all the other species of all the other animals combined. Insects are just incredibly diverse. And then we have fungi, the fifth kingdom. who are, like us, coming from a common ancestor, consumers. They're also heterotrophs. They need to get their food from outside sources. But they do it very differently than we do. We have a digestive tract. Fungi secrete enzymes into the environment, break down their food on the outside of their bodies, and then absorb it within themselves. So it's a very different strategy than we have. This is why fungi need moisture to work, because they need a semi-aqueous area that their enzymes can be in moisture and do their work. With the exception of yeast, fungi are multicellular, and they are mostly filamentous in nature. And so you can see here the filaments, the fibrous nature. Fungi don't have a body. They don't have a body plan like we would consider. You know, the animals are like the jellyfish, radially symmetrical. We are longitudinally symmetrical. We have organs, we have limbs, we have all our senses are in our head. You know, we have dedicated parts of our body that do specific things. Fungi aren't like that. Fungi are infinitely redundant. I can kill a person by stabbing them in the heart, right? I can kill a tree by girdling the cambium, but I can stab this guy as much as I want. It's not going to kill him because every part of the fungus is generally the same thing. So the way that they grow and the way that they live is very different from us, but they are our closest common ancestor compared to other forms of life. So what are some forms that fungi take? The forms I'm going to explain are not necessarily taxonomic groups. One thing that's interesting about fungi is they can often have multiple growth forms from the same organism. So one that we're very familiar with are yeasts, of course. So yeasts are single cellular. Some fungi are filamentous, but they have a yeast phase. So not all yeasts are dedicated single cells, but the one that we like to use the most, Saccharomyces cerevisiae, is the yeast that we use to ferment carbohydrates into alcohol, and that we like to use to raise bread, etc. They lend themselves very well to industrial processes because they're single cells. They reproduce like bacteria by budding, and so you can have a solution of yeast. You don't have to worry about it growing into a certain structure in order to work. So they're very useful for industrial processes, for example. Lichens. Lichens are a partnership between fungi and algae. And what this does is it turns the heterotroph into an autotroph by incorporating the autotrophs into themselves. I think it was about 15 years ago where it was discovered there's also a third partner, which is a yeast. So two different types of fungi that work together in a variety of different lichens. Primarily these guys, which are called lungworts, they're actually very ecologically important because some of these guys have cyanobacteria that fix nitrogen. So they're a major contributor of nitrogen in old growth forests, especially in the Pacific Northwest. But one thing that makes lichens really amazing is many lichens are highly xerotolerant, which means that they can handle very dry environments. They can survive for a very long time with maybe even one rainfall a year, things like that. And they're also considered pioneer species. If you have a major disturbance, Mount St. Helens was a great example of this, where the first colonists back after Mount St. Helens were lichens, and they kind of laid the groundwork. They started breaking things down and making things more available for others. Their acids work on rocks, for example, and liberate minerals. So they're some of the first colonists after a major disturbance. All right, so now we have the smuts and rust, which are obligate plant pathogens. Now the rusts generally don't hurt plants that much. Has anyone heard of apple cedar rust? That's a really common one. It grows on apple trees, and it doesn't hurt your crops, but it makes them look terrible, and no one wants to buy them, so it can really affect harvest. A lot of times the rusts don't hurt the plant, but these are all little ulcers that are releasing spores. These are structures that are meant to release spores. Usually the plants survive okay, but nobody wants them in their grain, right? The smuts are really interesting in that they hijack the plants. They live with the plant their entire life, and they secrete plant hormones that make the plant divert its sugars and nutrients to where it's growing. So they usually grow where the plant puts all of its nutrients anyway, in the flowers, in the fruits, in the seeds. And who here has had Huitlacoche? Very good, very good. About five people? That's fantastic. So Huitlacoche is a corn smut. And it tastes like sweet corn mushroom. It's because the plant, it's told the plant, send me all of your sugars. I am your most important seeds. You need to grow me instead. And then this is the result. Then eventually they act kind of like puffballs. They dry out, they release their spores. You don't want to eat them at that stage. But that's how they complete their life cycle. Molds. Molds generally are not a taxonomic group. They're more a presentation, a form that fungi take. Some fungi only exist in this microscopic form. So it's a little hard to even define mold, but in general, mold, all the structures in mold are microscopic. Now the colonies, of course, can get fairly large, but any of the individual structures remain microscopic. So you don't see mushrooms growing out of mold, for example. But because mold is a growth form, there are many fungi that create large reproductive structures like mushrooms and have this microscopic phase as well. What's the advantage? The advantage is very rapid growth and rapid spreading of spores. So it's about occupying territory. Most molds are producing spores in an asexual way. The spores are clones. So it doesn't give them an evolutionary advantage on the long term, but in the short term, it lets them occupy space very rapidly. And that's why it doesn't take terribly long in a damp environment for all these tasty materials like paperback gypsum to be colonized by mold. And finally, we have the mushrooms, which themselves are also not necessarily a taxonomic group. As we'll get to in a little bit, mushrooms are, like the mold structures here, reproductive structures only. The fungus is much broader and living in soil, in wood, in what we call the substrate where they're living. And they produce mushrooms as a means of reproducing, of spreading their spores. All mushrooms are sexual structures. They're the result of a sexual union, and the spores that they release are recombinant between both parents. So let's talk about the body of a fungus, the vegetative state, which we call mycelium. When a spore lands and germinates, it creates one filament as a starter. That's called a hypha. The hypha then branches out into more and more in a kind of an algorithmic fractal pattern like this. So 2 becomes 4, becomes 8, becomes 16, becomes 32, becomes 64, becomes 128, becomes 256, etc. I can go up to about 5,000, I think. Anyway, and so what does this do? Well, this gives it one of fungi's greatest advantages, greatly increases surface area. So fungi are excellent at penetrating, infiltrating, assimilating the substrate. Actually, I should say, I got that all to rhyme at one point. Maybe the next time I bring it up, I can get it all to rhyme again. Anyway, so I think the main thing that I want to express is that this is the form that fungi are taking. This is their body. This is how they live and grow, with the exception of some yeasts. The thing is that they're kind of hidden because we see what's on the surface, which is when they finally are ready to produce mushrooms. These are called primordia. They're early stage mushrooms growing. And so that's what we see. We see what's up on the surface. We don't see what's under the soil. We don't see what's in the log unless we break up the log or pull up the soil. And if you pull up the soil, you often will see these guys in the soil, right? You see what looks like cobwebs. And the soil smells like, has that earthy smell, right? That's called geosmin, which is made by a lot of fungi. And a wonderful bacteria called streptomyces. So here's just a really brief diagram that I made on the life cycle of fungi. So you start as a spore. When the spore germinates, it will form this mycelium. Depending on the strategy set of the fungi, they may enter this asexual cycle, which is a very rapid growing, fast occupation mode. And they also then, if they meet a partner that is sexually compatible, then they will what we call plasmogamize. They will combine their mycelium together the DNA from both parents, the chromosomes, enter new growth. And all new growth from that point on has the DNA from both parents. But it's not recombined yet, which is kind of interesting. Only when they finally form a fruiting structure, a fruiting body, is a recombination. It's basically right before the spores are made. And then the spores have the recombinant DNA from both parents. So this is a example of, here's your mycelium, here's your fruiting bodies, the mushrooms. And just to drive home the point that this is what the fungus is doing all year long, every day, growing like this. It produces fruiting bodies when it's ready to reproduce, and it wants to reproduce, spread its spores, and that's the main function it serves. So this is kind of analogous to fruits on plants, right? They're short-lived, ephemeral, and designed to spread their seeds. That's what mushrooms are. So that's a summary of fungi in general and their place in the tree of life. So how are mushrooms treasures to our ecosystem? Well, in one way, they have this amazing partnership, this symbiosis with other plants. So we call this symbiosis mycorrhizal. Myco being fungal, rhizal meaning roots. And so they have a connection, a physical connection with the roots of other plants. And they do this wonderful thing where they exchange nutrients between each other. I'll get to that in a second. The reason why I bring up trees and mushrooms is that most fungi that produce mushrooms that are mycorrhizal like this, they're doing it with trees. They don't do this with grasses. They don't do this with herbs. They do it with woody plants, mostly trees. So what's happening in this mycorrhizal relationship? Well, we have an autotroph and we have a heterotroph. So what does the heterotroph want? It wants food, and it doesn't want to work for it if it can get away with it, right? And what does the autotroph want? Well, it handles all the sunlight up here, and it handles all the breathing up here, but it needs water. And plants aren't nearly as good. You remember fungi's superpower is its ability to occupy space, its surface area. Penetration, assimilation, and I lost it, sorry. Anyway, so what happens is these guys are making carbohydrates, and they make more than they need, and they want help in their root system for taking up water and nutrients. There are some nutrients that plants are really bad at getting, like phosphorus. They're not very good at getting it, especially out of high clay soils, aluminosilicate soils. But these mycorrhizal fungi are actually really good at getting because of the surface area, getting lots of water and lots of these minerals out of the soil. So they exchange at the root system right here. The mycorrhizal fungi get fed, and the tree gets water, way more water and nutrients than it could get on its own. So I think this is a really great illustration of how this works. See this pine seedling? This is the root. Just these tiny little sections right here. That's it. All of the surface area, all of this root zone is fungal. So this is a great demonstration of how fungi greatly increase the surface area. Even the plant with the finest root, like if you pull up a grass, you got all these little roots, right? Doesn't even hold, it cannot compare to the amount of surface area these fungi can do. They're doing it at a microscopic level. So they greatly increase the ability of the plant to absorb water and nutrients. Conifers specifically, they've been doing this for so long. Conifers evolved before flowering plants. They've been doing this for so long, almost all of them don't even grow their own roots for absorbing things anymore. They only grow their roots, they grow it on their roots, we have what are called coralloid structures. And these are specifically to interface with the mycorrhizal fungi. And everything happens do you see these little nodules here? Everything's happening right there. That's where those exchanges are happening. If I clicked my finger and killed that fungus, the seedling would be dead within a year. And that would be true across the entire world. So how common is this? Incredibly common. The vast majority of plants have these relationships. Not all plants require it, but most plants have it. So 92% of all plant families have mycorrhizal relationships. Herbaceous plants, trees, everything. But again, when it comes to mushrooms generally only have the mycorrhizal fungi that make mushrooms, because they're decomposer mushrooms too, they're almost always connected with trees in some way. And this is why forests are a great place to find mushrooms. So who here has heard of the underground internet or the forest internet? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, almost a dozen, maybe a dozen people. When I first started giving this lecture, it was maybe one for years. So this is progress, right? How many people have seen Fantastic Fungi on Netflix or something like it, right? Right? So it's great to see that these, what used to be niche concepts and this niche part of biology is becoming much more mainstream. It's really great to see. So a couple of things, if you don't already know, a couple of the things that happen with these mycorrhizal networks is they're not just growing on one plant and sticking to themselves. They're all interconnected with the other mycorrhizal fungi. So they form a network, and things get shared across this network. Parent plants feed their seedlings by sending some of their carbohydrates through the network, and the seedlings get that. That's part of this relationship. Plants that are stressed or under attack by pests or pathogens, they release alert factors, they release resistance factors. It gets shared in the network, and plants all around it then start expressing resistance factors too, to protect themselves from whatever's attacking the tree or the plant that was attacked. So this underground internet is a real thing. It's verified, and it needs more study, of course, but it's fairly well studied now. It's pretty amazing. Who here has seen the Avatar movies, Avatar 1 and 2? So do you remember Awa, which is connected to everything, right? That Awa was inspired by this concept. All right, so the other thing that fungi do that are really important is they're recyclers. Especially with plants, they're powerful disassemblers, and they're part of the nutrient cycle. So I love showing examples of nurse logs. Who's heard of nurse logs? So this is a dead tree, and it's decomposing, but what else is it doing? It is a substrate for new plant growth. How's this happening? Because the log is being decomposed, those nutrients are being broken down and liberated so they're available for the other plants. This is how soil is built. And soil is being built on this nurse log here. Plus, nurse logs are great at retaining moisture because they're semi-rotting and that. So nurse logs, especially in the Pacific Northwest, serve a really important function in regeneration. So let's talk a little bit about what are fungi disassembling, especially when it comes to plants. Now, I guess I didn't bring this up. In evolutionary history, we have really good evidence for this. The first terrestrial land plants it was found that in their fossil records, they have these little nodules, these little connections, and we have good reason to think that the very first land plants colonized the land with the help of their mycorrhizal partners. So fungi and plants both came onto land together, and they've been co-evolving ever since as partners. So plants have two major structural molecules, cellulose and lignin. All plant cells, even single-celled algae, their cell wall is made of cellulose. And cellulose has some rigidity, it's reasonably pliant, but it's not strong enough to support large growth. You can't make branches just from cellulose. It's too flexible, it won't hold. So at a certain point in history, plants developed this molecule lignin, which gave it strength. Lignin is what gives wood its strength and its properties. In wood science, another name for wood is secondary xylem. Xylem is how plants move water up and down, primarily up to the leaves. These are called tracheids when they do that. It's the vascular system of the plant, and it's very strong. And every year it grows new growth to do this as the plant expands, and it abandons the old xylem, and that becomes wood. So why is this a big deal? Because lignin, not only is it strong, it's very hard to break down. And fungi are one of the only organisms on Earth that can do it. They use an enzyme called lignase to break down the lignin. And this is I'll get to why it's such a big deal in a moment. Who here has seen this in the forest? Yeah, this is called brown cubicle rot. This is when the fungi that are breaking down the wood are acting on the cellulose, but they don't have enough lignase to break down the lignin. And so the lignin stays, structure stays, the cellulose is eaten. We also have white stringy rot, which you probably have also seen. And you can just reach in, it's just, I mean, you can just pull out clumps of it. It has no structural strength. That's because it's been acted on by fungi that do make a lot of lignase and break down the lignin of the wood. So that's what makes fungi excellent at recycling in the nutrient cycle. They can penetrate, infiltrate, assimilate the substrate. I got it. Took 3 tries. Took 3 tries. And they have these wonderful enzymes. Fungi, one of the other fungi's other superpowers is they have all this cocktail of powerful enzymes that can decompose all kinds of organic compounds. And they have this very special and unique enzyme, lignase, that can break down wood. So I like to bring up this story. I think it's really valuable and gives us great perspective. What if this didn't happen? What if there wasn't lignase? What if fungi couldn't break down wood? What would the world look like? Like a tar pit? Well, a tar pit is mostly liquid. Well, how's the wood going to break down to the molecular level to actually be a liquid? Compression? All right. All that lignase that can't be molecularly broken down, it just remains, right? So if there was a time in Earth's history where this was the case, where lignin existed but could not be broken down. And we call that the Carboniferous period. It was about 360 million years ago. This, at the beginning of the Carboniferous, is when plants developed the ability to make wood. And so when you think about any ecosystem, ecosystems are driven by competition. What do plants compete for? They need the water in the soil, right? And they have mycorrhizal fungi to help them with that. They need light, and they need air. Well, in general, they can breathe the air no matter what. But light, they compete for. And how do you compete for light? You grow taller, and you grow out. You do what's called canopy domination. Plants couldn't grow this high until they finally developed the ability to make wood. And then the arms race was on, and we started to see trees in the landscape. The thing is, we got all this wood, but we don't have anything that can break down the wood. And so what happened? Trees fall and build up and fall and build up and fall and build up. And what are they taking with them? They're making carbohydrates. They're taking carbon dioxide out, but nothing is breaking down the lignin, so it stays. So what happens then? What happens is, very good, very good. 10 points for you, and you'll see. Here's the beginning of the Carboniferous. This is a logarithmic scale of carbon dioxide levels. Look what happens. So right now we have about 420 parts per million carbon dioxide, and it's rising every year, of course, as we know. At this time, before the Carboniferous, they were at about 1,000, 2,000, 2,500 part per million carbon dioxide. They went all the way down to 300 part per million carbon dioxide. So the carbon cycle was broken during the Carboniferous. Carbon was being sequestered and not liberated. So what this did also is it changed the ratio of oxygen to carbon dioxide. So who here has read stories, seen videos, what have you, of the ancient giant insects that lived before the dinosaurs, right? That was this time, because the ratio of oxygen to carbon dioxide was much, much higher in oxygen's favor. Insects don't breathe like we do. We breathe actively in and out. Insects don't do that. They absorb oxygen passively through little openings on the side of their body called sphericals. This is why you can kill a lot of insects with soap water, because it covers the sphericals and they can't breathe. The size of an insect, the size that an insect body can support, is based on how much oxygen is available. So when you had much higher oxygen available, you could have giant insects. So this is where you get the dragonflies and the millipedes as big as people, is in this time period. So as you said, what happens when all that carbon is not liberated? It just remains. And a large portion of the coal and a significant portion of the oil that we harvest comes from Carboniferous carbon from this period. But eventually, it took about 50 million years, but eventually we renewed the carbon cycle. And then we had a mass extinction event that killed everything. But eventually we liberated the carbon and renewed the carbon cycle. How did we do this? Fungi developed the enzyme lignase. It took them a long time, but they finally did it. So our world is the way it is today because fungi finally renewed this carbon cycle by developing this enzyme. They're kind of the heroes. They made life as we know it possible compared to the Carboniferous. So I like to tell that story. 2012. Right, yeah, and so when I first started giving these presentations, it was about 2012, so this was breaking information. Same thing with the forest internet, the underground internet was about that same time. So this is why fungi are very valuable to the ecosystem. They have this mutualism with lots of trees, lots of plants that let them absorb nutrients better, and they complete the nutrient cycle by breaking down wood. All right, so how are fungi treasures to us? Well, I imagine many of you came here because you're interested in wild mushrooms and foraging. So yeah, mushrooms are fun to eat. So I'll bet you all recognize white button mushrooms, and you probably all recognize portobellas. Who can tell me what's the difference between a white button and a portobello? Time and growth, yes. These are simply mature white button mushrooms that have been grown under light. They're melanized here. They're grown under light. So they're the same species. Same species. There are several different hybrids. If I name one species, there are definitely others. Agaricus bisporus is kind of the best known. But yeah, so the portobello are just mature button mushrooms. So porcinis, who all knows porcinis? Right, you've heard of porcinis. Steinpilz in German, Billy Grebe in Ukrainian and Russian. And so these are in a group called boletes, and we have lots of boletes in Arizona that come up during the mushroom season. Morels, how many people grew up hunting morels? I'm from South Dakota. It's like a state pastime in the spring. But now again, raise of hands, how many people know what morels are? Very good, yay. Sometimes you can get them in AJs. It's a seasonal thing. Sometimes you can get them in AJs, but it's like $80 a pound or something like that. Dried, $80. Right, yeah, it's dried, so it's not that bad. But morels are wonderful, and especially Midwest, Great Plains, very popular to hunt in the spring. Les truffes, truffles. Who here has had something with truffles, truffle oil, something like that? Very good. Has anyone here actually held a truffle, seen a truffle? Okay. They have this wonderful kind of pattern inside all these different nodules and that. Their strategy is not to release spores to the air, but instead they're aromatic and they want to be found by animals like pigs, animals that will dig them up. And their spores don't germinate. If I just took a truffle, ground it up, threw it in the soil, no truffles would grow. Their spores have to pass through the gastrointestinal tract of these animals in order, they have a coating on them. And the coating gets liberated through that process because they want to be moved. They want to be deposited, right? So we do have one edible truffle here in North America. It's the white truffle, and it's in the Pacific Northwest. We don't get these lovely black truffles. Those are European. Chanterelles, anyone? Very good. Yes, chanterelles are in Oregon. We have them here. We have them here in high elevation, like the montane forest, Mixicana fir, spruce fir. We have chanterelles here. In Russian, Lisichka obyknovenная, if anyone speaks Russian. Lisichka, little foxes. Yeah. Look at this happy guy, this happy Belgian. He's got a whole basket full of what looks like field bluets, and maybe one amanita here. But yeah, so we love to hunt mushrooms to eat. Foraging is really fun. Most cultures actually in the world, foraging is part of the culture. It's part of enjoyment. It's part of life. But for whatever reason, I haven't really researched this, but mushrooms get kind of a bad rap in Britain, in the UK, for whatever reason. And because America was founded by English colonies, that some of that kind of comes through in our culture too. Go ahead.
Are there more poisonous mushrooms in certain areas, and that's why it's so popular?
That may be a possibility, but you know, if I go to a stream almost anywhere in the world, especially northern hemisphere, I'm likely to find poison hemlock, right? That doesn't mean I can't enjoy the stream for other things. But yeah, so it may be possible that there are well, I wouldn't be surprised if poisonous mushrooms just got a bad rep because of some event and there was a spread of news like way back as part of the culture. I wouldn't be surprised at all. And you get these weird things too, like an important priest decides that there's a religious reason why mushrooms should be avoided, or these things do happen. I don't know the history of it. But so we kind of have a half and half here in America. Folks of certain lineages, of certain cultures, tend to look at mushrooms more negatively. They're poison, they're decay, and others. I have German and Polish descent, so we're very comfortable with mushrooms, and that's part of the culture of my ancestors. But I do like to bring up, if you go out to the forest just to look for things you can take home and you don't find anything you like, is your trip skunked? Did you have a bad trip? Right? So just putting 100% of your emphasis on just finding edible mushrooms kind of takes away from the rest of the enjoyment. I'm an ecologist. Ecologists are very much about observation and appreciation, not so much manipulation. And this is Galerina, a deadly poisonous mushroom, but I think it's quite beautiful, right? This is a bolete. It's Boletus santanus, I think. There's another one that it's harder to remember that it might be. But I found this guy, and some of them, one of the tests for whether a bolete is poisonous is if it has a strong reaction to being bruised. This is a very rapid blackening reaction when it's bruised. It's a diagnostic feature. And so this mushroom, which was very fun to find and identify, et cetera, and play with, cannot be eaten. So does that mean it's worthless and I shouldn't care and I should hate it? No, certainly not. This is an entoloma, which is not listed as edible, but they're quite pretty too. So I just like to bring up this idea of don't just limit yourself from what you can take away from the forest. Think, to really enjoy your time out there. You'll never get skunked. You'll never have a bad day if you focus on appreciation.
This whole talk, I'm very curious about emerging technologies.
Mm-hmm.
So are there, regardless of field, whether it's health, medicine, or other things, are there emerging technologies that are stemming from the study of?
Well, psilocybin's a great example of this. That's now become it's not regulated in Colorado, for example. And one of the obstacles to researching emergent biological technologies is, for example, cannabis. You can't really have a cannabis lab because it's still federally illegal. So it's very hard to do research when you're breaking federal law in order to do it, right? Same thing with psilocybin and that. There's lots that can be said about psilocybin, and I could, but I feel like it's a bit tangential, at least right now. Maybe at the end we can talk more about it. But psilocybin is really showing great promise for what we call neuroplasticity. It allows reconnection of neural pathways and even regeneration of damaged nerves. So it's great for PTSD, it's great for brain trauma, things like that. These are emerging medical technologies, and that I think we'll see more and more. When we truly understand how psilocybin works and how it helps with regeneration and neuroplasticity, that's definitely going to lead to new approaches to brain injury and PTSD and things like that.
Is there any way, if you're out going for mushrooms and you don't know what's poison or not, other than that this particular one could be bruised easily, is there any way to tell?
We have a whole section that's on identification that I'm getting to. Yeah, I'm sorry, I do have a fair amount of content, and I don't want to bring you over for those that need to leave. So we will hit on those, I promise. So I'd like to emphasize that even if you're not going for things for the table, or even if you don't find things for the table, you can still have a good time. So look at little Avid and little Cecily here. They don't want to eat these, right? They don't care about that, but they're beautiful and fun, and they're unique, they're different. They only show up at a certain time of year. They're unlike anything else that you see, right? So we all could stand to be a little bit more like little Avid and little Cecily, right? I like to talk about mushroom glasses or mushroom goggles a little. Humans are excellent at pattern recognition. It's one of our superpowers. And once you start seeing these things that you rarely see otherwise, then you start seeing more and more of them, and your brain adapts to be seeing the little glint of light, the light color in the background of the dark green, all of these things. And so getting into foraging, and especially mushroom foraging, is almost like it's almost like you've been cursed. You've spent your whole life looking forward and looking up and seeing the birds and the trees. Once you get into mushroom foraging, you're stuck. 50% of your time is down here, right? It's not any less interesting, but it does change the way that you interact with your environment. So look at all these beauties. Almost nothing up here is edible. Almost nothing. Several of these are poisonous. But would you be unhappy to only find these if you went on a foray, right? I think we all would find a lot of these very interesting to find. Yeah, bioluminescent mycena up here. Yeah. So I found maybe about 30% of these guys. Some of these are from other countries, but yeah, some of these grow in Arizona. Scaly vase chanterelle is here. We have quite a few of these corals that grow here. Turkey tails grow here. I don't know if I've seen this specific entoloma, but some of these, this is Floccularia straminipes, I think. We have these here. So yeah, don't limit yourself. Don't limit yourself for what you can take away and bring home. All right, so now let's talk about what are the characteristics of mushrooms. What are you looking for? How do you identify them? So I have a section in my presentation for this, but I kind of like going to my website for this instead, because I have kind of a dedicated section. Uh-oh. Do I have to minimize the presentation in order to do that? Let's see. I thought it was what I see on the screen is what you see up there. I might have to close the whole thing. Let's see. I hit the X.
Do mushrooms grow in the ocean?
There is one aquatic mushroom that was found, I think, in Crater Lake. It's a Sathrella, if I remember right. But in the ocean, no. But there are fungi in the ocean. It's not even recognizing when I hit the X to stop it. That's so weird. I know, I hit the, all right, there we go. All right, so hopefully, yay. All right, let's go to full screen. So in the handouts that I have, here we go. In the handouts that I have, I do have a reference to my website, which I call the Arizona Mushrooms Primer. And it goes into everything we've talked about, the kingdoms, taxonomy, ecological roles, but then I also have a large section on identification. So identifying mushrooms is actually not that different from identifying plants. You're looking at the characteristic features, the features that distinguish one from another. And the cool thing about fungi is lots of these features are taxonomically, evolutionarily conserved, which means if it has a certain feature, all members of that family likely have that feature. So it can be very helpful for figuring out what you're looking at. So when you're looking at a mushroom, you're looking at the cap. What color is it? What texture is it? Does it have these striations? Some mushrooms are known for the gills leaving lines on the cap. These are called striations. The gills, does it have gills? Does it have pores? Does it have teeth? How do the gills connect to the stem? All of these things within a family or within a genus are all the same. So it's very useful taxonomically. Does it have a ring, or what we call an annulus or a skirt? Does it have a sac at the base? Is it growing straight? Is it offset or centered on the cap? All of these are features that you're looking for when you identify mushrooms. So let's have a quick look at the gills. So we distinguish, trust me, I'm not going into great detail, but we recognize that a gill attachment is taxonomically important. This is an agaricus. If you go to the store and look at a portabella, the gills stop before they reach the stem. That's a characteristic feature. Agaricus has a skirt. You'll see these on the portabellas, right? I know it's an agaricus because it has a skirt, the gills are free, and the gills turn chocolate brown where when they're young, they're pink. If you look at a white button, it has pink gills. If you look at a portabella, it has brown gills. That's because the spores are brown, chocolate brown. As the spores mature, it changes the color of the gills. So here are some examples of different types of gills. And then we have some that are pores and not gills. So we have a whole group of wood decomposers called polypores that form big woody structures on the side of trees. This is from what we call a bolete. So porcinis are in this group, bolete. They have pores instead of gills. Here's teeth. Who's heard of hedgehog mushrooms? Popular in the Pacific Northwest. You go to Pike's Place Market, you'll find hedgehogs for sale. So these are all ways of getting as much surface area as possible for the spores you're going to release. The spores are being made in the gills and the pores and released. Caps, all these different shapes, and there's a name for each one of them. right? Scaly caps, warty caps. So I'll get to, this is an amanita. I'll get to amanitas a little bit later, and we'll talk about what's happening on the cap and why that's a thing for amanita. Striation, can you see the lines? Let's see. Because of time, I did bring one little demo, and I forgot to start in the beginning. Has anyone heard of Earth stars? Okay, what I've got, I'm sorry, it's the size of a quarter, and it's a big room. But this is an Earth star all dried up. And it doesn't look like much right now. But Earth stars are really special in that they have a reaction when they're exposed to water. It's a little slow, so maybe at the end of the presentation I'll show you what happens when it's done. Those of you in the front row may notice it changing over time. So slimy caps. Slimy caps are really interesting, and we have a really common bolete here in Arizona called slippery jack. Mushrooms can't release their spores if they're waterlogged. They need a certain amount of humidity in the spores because they rely on droplets forming to kick the spores out. So they have all these strategies for protecting the spore-bearing area from rain. And slime is a great way of doing it because the water just runs right off and doesn't get into, see how all these are like, what would you call it? Umbrellas, right? In fact, this is Lepiota, it's called a parasol mushroom for that reason. Rings and skirts. Here's a ring. So we have one here in the valley, a big white greenish one called green-gilled Lepiota, if I remember right. Chlorophyllum is the taxonomic name. It has a ring that freely slides up and down the stem. That's one of the things you can check to identify it. Skirts, like this guy's an amanita. The skirt is attached to the top. This is a group of mushrooms, a very common group called Cortinarias, and they have what's called a cortina. This is like a cobweb. And you can even tell what color the spores are because the spores are all caught in this cobweb here. Stems. This is called a scaber stalk. See the texture on here? Those are called scabers. Here's scaly stems, scaly stem foliota, scaly foliota. Reticulated stem, you might not be able to see it, but these look like little capillaries. This is, Porcini has this. Wiry stems, tiny little wires. Stems that change in texture between top and bottom. This is called woolly-footed colibia. Woolly-footed, you see the mycelium that grows there? So amanitas have this characteristic of having a sac at the base. And so you look for that sac. There are some deadly poisonous amanitas, so amanitas are one that you have to become familiar with. And you look for that. Color changes, I already showed you some, right? Color changes. There are poisonous and good agaricus, like the portobello. Some of the poisonous agaricus have a yellow staining reaction and have an odor to them that you check, things like that. There's a group called Lactarius that actually bleeds latex if it gets damaged. And so that's a diagnostic test, right? Odor and taste. The poisonous agaricus smell like asphalt. The good ones smell like mushroom or smell like almond, for example. Taste. Rustulas, which I'll get to in a little bit, the way you tell the difference between a poisonous and a safe rustula is you do a nibble test on the cap, and if it's peppery, it's got the compound that will sicken you. Bitter tasting. This is a really fun one. This is a coral club mushroom called a sweet club, and it has a sugar alcohol in it called trehalose that tastes sweet, for example. Spore color is a really big deal. So taking spore prints is part of identification, figuring out the color of the spores. This is because spore color is very strongly taxonomically conserved. I can tell you that members of Tricholomaceae have pale colored spores. I can tell you that members of Strophareaceae have purple to black colored spores. Psilocybin is in that group, for example. So this is a Stropharia. It has black spore print. And you can see that sometimes you don't have to actually take the cap and put it on paper. You can just see it in the field, for example. Here's a white spored Clitosabe. And you can see the imprint that it left on the ground here from all its spores. Growth patterns, are they solitary? A lot of mycorrhizals are solitary. They don't grow in clusters because they just come up wherever they want around the tree. Clustered mushrooms are usually decomposers because they're acting in one spot. Their food source is right there. And so everything they grow is going to be right there. Troops, so many in an area but not in a cluster. Scattered troops, like these marasmias. Fairy rings. It's a little harder to see fairy rings in the forest, but you can see them, and in grasses, you can definitely see them. These are agaricus. These are field mushrooms here. The mushrooms are all gone. They probably mowed over them, sadly. This was at a school. This was a news article. Amazingly still exists. It's like 15 years old now. But these are all fairy rings from agaricus mushrooms. And then what is it growing on? Is it growing on wood? Is it growing on a tree? Is it growing on the edge of where other things are decomposing wood, and maybe it's a secondary decomposer? These are inky caps. They're secondary decomposers. They can't act on fresh wood, but they rely on these scaly foliotas to break the wood down, and then they eat the semi-broken down wood. Neef and Little, that was great. Leaf and needle litter, for example. Are they just out in the field? And are they just coming up from soil with nothing else? Mycorrhizals, of course, are attached to the roots of the trees, so they're usually in the drip line of the canopy of the tree, and they're just coming up from the soil because that's where they live. So when you identify, I highly recommend, first off, you need guides. Secondly, when you're going to find the majority of mushrooms you find, you're not going to know what they are, right? So you can just skip by them and focus on just the ones that you know, or you can learn. You can learn new ones. And what you do is you take notes. You do everything we just talked about. Your field guide is going to say cap, these features, stem, these features, base, these features. And you write down these features in the field. If you bring your field guide into the field, during the monsoon season, it rains at noon. And it rains for like 3 hours or something like that. You're going to waste your time if you take out your field guide for every little thing that you find. Write notes, take photos, figure this stuff out at home. That's my recommendation. Field photography, photos worth 1,000 words. When you take photos, you want as many features as possible. Don't just take a picture of the cap. That's 10% of all the information it has for you. You want to see the gill attachment. You want to see, does it have a ring or not? All these different things. Get those in your photos. I highly recommend that you do notes in photography if you're going to try, if you don't want to waste all of your field time. So now let's go back. I actually could do the rest of this just off the website, but it just needs to load. It's on the USB stick. Or I have to do that. Oh man, I'm way back. All right, just a second. I'm going to go through this very quickly, just the different types, because some of this is redundant. Okay, so we talked about gills, pores, teeth, puffballs, which are none of these things. Earth stars. So our Earth star is half done now. Are you able to see it? See it?
It's an alien.
Give it time. It's not done. There's a very specific reason why Earth stars do what they do. Say again?
It was dry.
It was dry. And this one is Astrius hygrometricus, which means water measuring. It's great for demos because it will dry up. You wet it, it opens, it will dry up. You can do this many times, so you can get away with that. Here it is. Here he is, fully done. That's our guy, Astrius hygrometricus. So I guess since I have this nice picture up here, I'll just explain. So like I said, fungi that want to release their spores to the air, they can't do it if they're waterlogged. It has to be dry enough that the spores can be released. So if this puffball is completely saturated with water from the rain and that, none of those spores are getting out. So this is specifically designed, you see, it's hard to see, but it has a little hole on the top. When a raindrop hits that, spores come up. Like a little tap it. And of course, you can do it 1,000 times harder if you just step on it. You create a giant cloud of spores. But the Earth star, when it's wet, it raises up on this outer membrane that's called a peridium. And it raises it off the ground so that pooling water doesn't soak it and waterlog it. That's the strategy for Earth stars. Oyster mushrooms, which are a fan shape. I'm kind of showing the different forms they take. So I'm sure many of you, raise a hands, oyster mushrooms, you know them, have eaten them. Sometimes they're in our common grocery stores. Occasionally you see them in our common grocery stores. I think AJ's usually has them too. So they're more fan-shaped, right? This is the jack-o'-lantern Omphalodus luteans, which has a very weak glow-in-the-dark bioluminescence, a greenish bioluminescence. Turkey tails, we call these bracket fungi, where they form just little shelf-like structures. Here are what we call conks, C-O-N-K. It's a dangerous word to say quickly. Where they form these are perennial. These are multiple years of growth. Every year they add a little bit more during their growing season. And one of them is called the artist conch because it's white, but when you scratch it, it darkens, and it stays that way. It's permanent at that point. So people will do artwork with them, for example. This is called witch's butter. It's a jelly fungus that, what does it have the texture of? Like gummy bears, gummy worms. It's fairly tough, actually, but it has this gelatinous, and when it's dry, you will not see it. Almost 90%, like 95% of its mass is water that's absorbed. Coral mushrooms, which is my daughter's favorite. This is Cecily's favorite. Cauliflower mushroom. This is called spiraceus, and everybody tries to find the spiraceus. There's usually one for every 5 miles. You find them under dog fur usually, and they're really nice. They're great for eating. Okay, so let's talk about some common Arizona mushrooms, ones that you're likely to find if you go out. Not all of these are necessarily edible, but these are the ones you're going to find a lot. So we talked about boletes. They have a classic mushroom shape, but they have pores instead of gills. This is called a slippery jack, and it is a bolete. There are different groups within the boletes. This is the genus Suillus, which are the slippery jacks. We call it that because when their cap gets wet, it becomes slimy and it sheds water very easily. So you kind of recognize that in the field. You see how it's kind of slimy. They have what are called glandular dots on the stem, if you can see these. That's part of their characteristic feature of how you differentiate them from the porcinis, for example. Very common, they're mycorrhizal. They occur mostly with pine, so you find them in ponderosa. They are edible. Not everybody likes them because their texture is fairly soft, like a marshmallow, even when they're cooked. But the young ones, I know folks who like to pickle them. They're really good pickled, and they're much more firm when they're young like that. There's a special rule, you have to peel the cap off because that slimy peel can cause problems. Russulas, these are really common. You'll see them pretty much everywhere. Some russulas are edible, some are not. The test for russulas edibility, it's not a guarantee. None of these tests, especially a perceptual test like that, if it's an older mushroom or it's a really dried out mushroom, it might not have as strong of a flavor, but a peppery taste distinguishes sickening. This looks like Russula emetica, which is a sickening russula. This looks like, I'm not sure the species if it even says, but they're very common. Russulas are interesting because they actually can become lobster mushrooms. Who's heard of lobster mushrooms? I'll show you lobster mushrooms a little bit later. Lobster mushrooms are actually a parasitized russula. Another fungus takes it over. I'll explain that in a little bit. But you're going to find these a lot. They tend to have a little dip in the top, and their texture, they don't have a skirt or anything like that. Their texture is really interesting. You know how pears are kind of granular? You can kind of even break a pear in half. Most mushrooms are fibrous. If you try to break them apart, they'll just kind of tear, and you'll see the fibers there. And these guys break like chalk. They kind of have this consistency like a pear. They have special cells called spherocysts that keep them from forming the fibrous growth. And so the test for russulas is you break it like chalk, or you give it to a kid and have them throw it against the tree, and it just becomes bits. All right, so turkey tails, very common. There are many different types of bracket-type fungi. You'll find many different types. Turkey tails are great because they have medicinal uses. It's pretty well researched that they have some immune-boosting abilities. If you get like the mushroom additives that you can put in your drinks or food, you get mushroom coffee, mushroom tea, they usually have turkey tail in it. And here's the conchs again. This is Ganoderma. This is not the artist conch, but it's closer. Ganodermas tend to grow out flat. where this guy is Fomitopsis, and it tends to grow more like a hoof. So these guys every year, they grow down a little bit and out a little bit. Ganodermas tend to just grow out every year. Ganodermas are another one. Has anyone heard of reishi tea? That's a Ganoderma, and that's often used in the teas and coffees and that. Amanitas. You're going to find a lot of amanitas out there, and so it's important to recognize them because we do have some deadly amanitas in Arizona. It's important to recognize them. The nice thing is we have a nice edible amanita here that doesn't look like any of the others. It's pretty easy to distinguish, and that's this guy. A lot of people call this guy a Caesar's amanita. We've learned that it's not the same. Caesar's amanita is a European species. We have what's called Amanita cochiciana, which is it's a Rocky Mountain, southern Rocky Mountains, Colorado, Arizona. But it's edible, and it's all orange. It can have a whiter stem, but all these orange features, and it has this really strong membrane around it when it grows. All amanitas do this. I'll go to the next page and show you. But who recognizes this guy? We have these guys here. Yeah, does anybody know the name for him? Go ahead. Go ahead. Yeah, you're right. You're totally right. Yeah, absolutely. Amanita muscaria. That's right. So our version, you see how these are a little bit yellowish and not as bright red and pure white? So our variety is a little bit more yellow than the Eastern variety, and the toxins in it, it has no or very weak hallucinogenic, but it does have some sickening toxins, but they're weaker than they are in the Eastern variety. So some people prepare these with a double boiling procedure, and you can eat them. They're actually quite good that way, but I don't recommend it because there are toxins in it. So that's completely up to you. So how do you identify amanitas? Well, they start off in this big membrane that protects them. And this is one of the main features you look for. You look for the sac at the base of the amanita. In the case of like these, the Caesars, the sac is so strong that they just pop right up out of the sac and none of it comes with them. In the Muscaria group, it's very fragile. The membrane is very fragile, and so it leaves little bits behind. That's what this is. This is that membrane around as it's coming up. So these are the things that you look for amanitas, is you look for that sac, you look for a skirt. There are a group of amanitas called grisettes. Here's an example that don't have skirts, but most of the amanitas do have skirts. You look for straight stems. They tend not to grow in an awkward fashion. Stems that hit the center of the cap. They usually have striation, which I'm not sure. Maybe you might be able to see a little bit of it there. But you see the lines where the gills hit the cap. And let's see, so this artist needs to be slapped because he has the gills coming down the stem. That's not true. All amanitas have free gills. You see this little boundary here? So the gills stop before they reach the stem, for example. This is the potassium hydroxide test. They turn yellow with potassium hydroxide. Most of you probably don't have a little potassium hydroxide dropper like I do. Amateur mycologists have iron salts, potassium hydroxide. There's about 4 different reagents that you bring with you. And good field guides actually talk about what their reactions are. So any of you who intend to forage for mushrooms, I highly recommend you spend some time learning amanitas, the characteristics of amanitas, because we do have deadly poisonous amanitas here. We have, I'm not sure if we have death cap, but we definitely have destroying angel, which is an all-white amanita.
Really quick, how long does it take to get from young toadstool to on the very right?
Between 3 days and a week. So one of the things that's interesting, I'd have to page all the way back to the beginning, but when mushrooms start to form, most of their features are already formed before they even come up out of the ground. It's like a cluster of tissue, but it's already differentiated. The primordia do that. And then what happens is the fungus basically just pumps it full of water and other things that are in it and inflates it, basically. So there's not much difference in biomass between this and this. It's just how much water is in it. How much of it has it swollen up into its shape. So that means that you can get overnight, you can see mushrooms in some cases. That's why they can grow so fast. So for the last section here, I have these 4 mushrooms that I think are easy to identify. They don't have poisonous lookalikes. They're a good 4 starter mushrooms for anyone who wants to forage. So I'll start off with puffballs. Here's little Cecily with her with her giant puffballs. Those are Clavacia. So it's hard to find these. Sometimes you find them. They tend to be found in fields or like on roadsides and things like that. These puffballs here are what you're going to see way more of. These are our group called Lycoperdon. And they often are found on rotting wood with moss, so semi-already decomposed wood. Some of them actually just grow on the ground. I've found them on the slopes up at the ski lift in Flagstaff, for example. They tend to be all over the slopes there. But then in the woods, you look at semi-rotten logs with moss growing on them. So puffballs, when they are young and just forming here, they're undifferentiated inside. It's just mushroom. And that's when you pick them and eat them. Eventually, then, those become spores. And then that's absolutely disgusting. And it actually is sickening also if you ate that. So what you do with a puffball is you cut it and check it, because you do not want one of these, right? You don't want that in your puffball. That's why you check all your puffballs. And you check to see if it's all white. If it's yellowing or browning, then it's at a point where it might start to be sickening. So you catch them when they're early. The aspen bolete. This is related to porcinis. It's a bolete. It's a different genus, but it's in the bolete group, so it has pores instead of gills. It only grows with aspen. This is one of the interesting things. It's a mycorrhizal. All right, so actually, let's see. This is decomposer. This is mycorrhizal. A lot of the mycorrhizal mushroom-producing fungi have a very limited group of trees that they will grow with. It can even be down to a single species of tree. The aspen bolete only grows with aspens. And we also call this the scaber stalk. It's not as obvious in this photo, but can you see the texture here? So these, the glandular dots are just kind of, they're visually there, but there's not much topography. These are actually rough. You can actually see these roughen. Light brown to kind of an olive brown cap, cream-colored pores, and then this kind of cream-colored stem with the scabers. It has a color reaction if it's bruised. It very slowly becomes dark blue over time. But this is a really good edible. It's about as equal to a porcini in edibility. Shaggy manes. Who knows shaggy manes? Who's seen them, right? So down here in the valley, we don't have shaggy manes. We have what's called desert shaggy manes. They look very similar, but they're actually stalked puffballs. So if you play around with them, what you'll see is they grow up, they look like this white lollipop type thing, but then the outer part becomes very fragile and papery, and then if they get disturbed or broken at all, then spores just pour out. So they're actually a puffball down here. This is in a group called inky caps. It's a true mushroom. The inky caps have this interesting strategy. They don't release their spores to the air. Instead, they turn to this black ink when they reach maturity, and the spores just fall on the ground and they wash away. The rain carries them away. Insects that walk across carry the spores somewhere else. So that's their strategy. They don't use an air strategy to spread. Shaggy manes are great if you collect them when they're young in this stage. Even if you keep them in your refrigerator, they will start to turn to black ink within like a day or two. So you have to collect them and prepare them fairly quickly. They're edible in all stages. The ink is edible. Some folks will use it as a flavorant because it has a mushroom flavor, and I've heard it's quite strong. But I think most people would probably eat this stage and not this stage, right? But they're fairly fragile, and they have a lot of water in them. It's good to sweat them in the pan first, but you do have to prepare them the day of collection. Fortunately, there isn't anything else that looks like it. And the desert shaggy manes here, when they're young, are edible. And especially in Sonora in Mexico, they're collected and used as edible mushrooms. And finally, we have the lobster mushroom. So who has actually found and handled lobster mushrooms? They're one of the more common ones. All right, very good, very good. Who recognizes lobsters, has heard of them, maybe on cooking shows or something like that? All right, so lobster mushrooms, we talked about russula. So russula are everywhere. They're one of the most common mushrooms you're going to find. The lobster mushroom is after this other fungus. It's a parasite called Hyphomyces. infects the russula. Russulas tend to develop underground. That's why they often are covered with dirt, and they're very dirty, right? So this often happens underground when they're forming, and they get parasitized. Hyphomyces completely takes over and replaces the tissues with its own. So there are even, the most common russula that gets lobsterized is this Russula brevipes, this white one, which is edible. But there are even sickening russulas that, because they get completely replaced, they're made edible. So lobsters are really safe. If you see this orange, if it's a tough orange, it looks like this, then you're pretty much good to go. There are a few other hyphomyces, but they're different colors. They go after different mushrooms. So the orange lobster mushrooms are always safe, unless they're like half rotten or something like that. But so they're really good. They're very common because russulas are very common. I'd say that they're probably the most commonly found edible mushroom in the Flagstaff area, for example. All right, so that sums it up. Why are mushrooms treasures? Well, they're treasures to the ecosystem because they really help the plants get the nutrients they need, and they break down dead vegetation. They complete the carbon cycle. Why are they valuable to us? Well, because they're fun. They're good to eat, and they're fun to find. Thank you.
Thank you very much.
Thank you.
Do you have a couple minutes for a few questions?
Yeah, I'm happy to take questions.
Okay.
But yeah, of course, of course.
So half the hands went up in the room. Okay, so we are running a little over time, so I'd like to keep your questions brief, to the point, and again, we've got less than 30 minutes until a board meeting, but we can run past.
Good.
If we need
I'll
to.
do my best to keep my answers brief.
So
to
we're going
the
to
point
start
too.
here, and then we're going to go over
Okay.
here.
Before we start, though, if I may, just for a moment, I do want to put a plug in for the Arizona Mushroom Club. We do have a mushroom club in Arizona. It's the Mushroom Society. Everyone wanted it to be Mycological Society, but there were legal reasons why they couldn't do it. It's a liability thing. But if you want guided forays, if you want to go out with experienced people, if you want to practice amateur mycology, we have a lot of folks who are interested academically, where we identify mushrooms and we submit them to herbariums, and we have found new species here. The Arizona Mushroom Society is where to go. They have a website. It's in the handouts that I gave. For example, I highly encourage you to join, $20 a year, and you'll get guided tours. Here's an example of an after foray where we lay everything out and we explain what everything is. For example, the lady at the end of this table here is my mycology professor that was part of my thesis committee. And so we have highly qualified people in the group that give guided forays where you can learn a lot. Go ahead, please.
Yeah, I was just curious, are mushrooms an important food source for animals that live in forests?
They are. So we have, I'm going to butcher the word, but I think it's called mycotrophism, if I remember right. There's a better word for it. Anyway, we have some unique squirrels in Arizona. The Abert squirrel, which is northern Arizona, and then we have the Kaibab squirrel on the northern Kaibab plateau. Something like 80% of everything they eat after August up through the middle of the winter are truffles and russulas that they collect and put in their caches. So a very high percentage of their food are mushrooms and truffles at that certain time of the year. And mushrooms provide lots of ecosystem services. Lots of insects lay their eggs, and part of their life cycle includes the mushrooms themselves. And of course, there are a lot of tree pathogens, like the bark beetles. Bark beetles don't eat the wood. Their larvae eat a fungus that the adults bring in with them when they burrow in and lay their eggs. And the fungus grows in the bark, and it's the fungus that kills the tree. It's called blue stain fungus, Ophiostoma, if I remember right. And that's what kills trees. It's not the bark beetles, it's the fungus that they introduce that clogs all the cambium, and then the larvae eat that. So there are lots. This is one of the things that makes mycology really interesting from an ecological standpoint. There are all these relationships with all these other forms of life. The ecosystem truly is bound together and interconnected because of fungi.
Cool.
Go ahead.
So you talked about parent trees sending nutrients to their seedlings through the mycorrhizal network. Are they just kind of spamming nutrients into that network, hoping they get to the seedlings, or is there an actual transfer of information that they know which plants are their seedlings, and what do we know about how that works?
I think it's more general than that. This is not something that I've studied deeply enough to be able to cite a paper. I'm sorry. I wish I could. We all have to pick our battles. But to my understanding, it's not as targeted as that. It's not the nutrients that the tree shares with the mycorrhizal network is not intended for a specific destination, and it's not carried to a specific destination intentionally, to my understanding. I think it's just kind of all part of the network and kind of diffuses out that way.
Maybe a simple question. What is the deadliest mushroom?
Yeah, probably the death cap and the destroying angel, which are both amanitas. Amanitas phalloides and Amanita ocratia, if I remember right. And then we also have another one. It's an LBM, what we call little brown mushrooms. Birders have LBJs, little brown jobs, little finches, and all that. So we have there's a whole group of mushrooms that are hard to identify. They're just little brown mushrooms. One is called Galerina, if I remember right, Galerina marginatus. We could look that up. I had a picture like halfway through when I said edibility versus fun. Galerinas are very dangerous, and because they look like a lot of other mushrooms, I'm on the poison control call list, and several other mycologists in the state are on this call list. And we get these horrible, blurry little photos of things that you can barely tell. And it's very hard to rule out Galerina because it's just a little brown mushroom. It has some very specific features, but it means that the person who took the picture and the mushroom itself has to be in a good enough condition to help you. But yeah, and these all have what are amytin poisons. And if I was a little more fresh on it, I could tell you the specific toxins that are in amanitas that kill. What they do is they get in your liver, and they interfere with your ability to make ribosomes. And ribosomes transcribe the DNA from the nucleus and make proteins. Ribosomes are in the endoplasmic reticulum, the rough endoplasmic reticulum. Anyway, it inhibits your cell's ability to make those. And so you lose the ability to make proteins, and you basically you can't make the proteins that you need to live, and you die. It takes about 2 weeks. We have those here. Yeah. This is why I highly encourage people, one, not to pick random little brown mushrooms that you can't identify and wouldn't be good anyway, but two, know your amanitas, know what amanitas are, because we do have a deadly amanita here. Go ahead, Ken. Oh, sorry, go ahead.
Since they don't occur in heterosexual pairs, how does Noah deal with mushrooms on the ark?
I only heard half of that, Ken. Could you say it again? I'll repeat your question. I just need to.
Since they don't occur in heterosexual pairs, how did Noah deal with mushrooms on the ark?
Oh, that's a great question. I would love to have a witty answer, but I've already expended most of my mental power on this presentation.
When did you have brought in the ark?
I'm not going to talk about the ark because, as we all know, there's no point in being realistic in this conversation. Anyway, sorry, we have a mic'd guest here that I'm happy to answer.
So I can't grow things. I'm terrible at plants. But you were mentioning earlier that there's that relationship. So is there a benefit to adding certain like fungi to your gardens and stuff to
Absolutely.
Okay.
Yes, absolutely.
Tell me more.
You can get mycorrhizal soil amendments that you can order from nurseries and from other sites. Paul Stamets has his Fungi Perfecti website where you can order all kinds of things. Doing mycorrhizal amendments to your garden, to your trees, etc., is definitely beneficial, especially to new plants, and especially if you're not growing in native soils where these plants naturally occur. So yes.
Fantastic. We have time for at least one more question. If you wouldn't mind sticking around for after we're done with the presentation.
Happy to.
While we're getting set up for the board meeting for further questions, we have one final question, and then we're going to end our recording, and then we'll go from there.
Okay.
Thank you, Eric. Tell me, plants and animals are suffering from mass extinctions. Are we seeing any of that as a result of climate change and human occupation of the earth on fungi?
Yeah, fungi also suffer from habitat loss, for example. They're adapted to specific environments. There are, like many other forms of life, there are generalists. Like when we introduced mice to Australia, they went wild. It wasn't a place that they grew up in, but they are generalists and they were able to adapt to it. You see that in all forms of life, but you also have lots of specialists. And it's the specialists that suffer the most when you see changes in an environment. Loss of habitat, shifts in climate, etc. We're seeing a strong drying out of many areas in the Amazon rainforest right now, for example, even though the new president has done a great job of reducing the amount of deforestation. They're dealing with all the other impacts, and the forests are forever changed now. So one of the things that's interesting about fungi is I think we only have documented something like 10,000 species. It's expected there could be 1 million to 10 million species undiscovered. So we don't even know how many are dying out or being excluded. But yeah, fungi are also vulnerable to all the things that these other forms of life are when it comes to extinction and population loss.
Fantastic. If you would please give a warm thank you, applause for our amazing speaker today. Thank you so much. We greatly appreciate it.
Thank you.
All right, so as we're wrapping up here, I want to say thank you so everybody so much for attending today. If you enjoyed yourself, please considering becoming a member, volunteering, coming up with ideas with events that you might want to see. And also, there is a lot of food in the back that we cannot put back in the refrigerator, so if you could take it with you. And again, we do have a board meeting here in about less than 20 minutes. So if you'd like to stick around for that, that'd be great too. Live humanism every day. Thank you, everybody, so much.